Step 1. Transcription.
Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. Transcription is like photocopying papers. You need to copy the right papers. You choose what you need to use at that moment. The process of transcription starts with the DNA, the code for all proteins. A portion of the DNA unwinds or unzips itself to use as a template for the mRNA. Nucleotides of mRNA match themselves up with the nucleotides of DNA. The nucleotides are the four bases adenine, thymine, guanine, and cytosine, as well as one other, uracil. The cytosine and guanine and the adenine and thymine of the DNA match up. There's one exception though. Whenever there is an adenine on the DNA, a uracil will match up with it on the mRNA.
This process is only used on the portion of DNA that codes for the protein the cell needs right at the moment. So, for whatever protein is needed, the DNA will only unravel and allow the mRNA to read that exact segment.
Only around 2% of our DNA is actually used. The newly made mRNA will now leave the nucleus through the nuclear pores. This is now the beginning of translation.
Step 2.
That's a better deal.
Translation.
Translation is the next and final process in protein synthesis. This is the process in which cellular ribosomes create proteins.
Translation occurs in the cytoplasm. Translation has to translate the message inside the nucleotide sequence of mRNA to a specific amino acid sequence. The mRNA comes out of the nucleus and immediately attaches to a ribosome. The mRNA will be read 3 bases at a time. Each group of 3 are called codons. There is an anticodon which is connected to the tRNA. A start codon will be read, and the first amino acid will be brought in by the tRNA. The anticodon and the codon of the mRNA match up. After they connect, the next tRNA comes in and matches up with the codon. The two amino acids will then form a peptide bond. The first tRNA will detach, and the mRNA will move to allow the next tRNA to come. And finally, the protein will continue to grow until it meets a stop codon. Once the last codon is read, the protein will be formed and then will finish folding to become functional.